Welcome to the BioInsights, where science comes alive.
Today, we're taking a journey deep inside the most complex, highly efficient, and honestly beautifully chaotic microscopic factory on Earth, the human body. Every single second, in every single cell of your body, an untold number of chemical reactions are just firing off in perfect harmony. And they're doing this to keep you alive, keep you thinking, and keep you moving. But, how does this factory actually operate? What is the machinery that's really running the show? Well, here's the incredible paradox of biology. Think about it. If you were to leave a simple solution of sugar sitting in a jar, sure, it would eventually break down. But that natural chemical reaction is incredibly slow. I mean, so slow, it's barely measurable. For life to even exist, we obviously can't wait years to digest a single meal or build just one muscle fiber. Your body needs a special mechanism to speed these reactions up by literally millions of times. And it has to do all of that without raising your internal temperature to the boiling point. Okay, let's dive into Section 1, Meet the Biological Catalysts. To solve this crazy problem of speed, biology relies on some highly specific machinery. And this brings us to the absolute stars of our explainer, enzymes. An enzyme is a biological catalyst. Now, in chemistry, a catalyst is basically just something that lowers the activation energy required for a reaction to take place. It just makes it way easier and way faster for that reaction to happen. In our bodies, these catalysts are usually globular proteins, which means they're these long chains of amino acids that are intricately folded into very complex 3D shapes. You can almost always spot them by their name, too, because they typically end in ase. Think lactase, sucrase, or protease. They govern basically all the biochemistry happening inside you right now. Now, in this vast biochemical factory, enzymes generally have one of two main jobs. On the one hand, we've got anabolic reactions. These are your builders. They take smaller molecules and bind them together, using energy to build complex structures, like synthesizing protein to build your muscles. And on the other hand, we have catabolic reactions. These are the breakers. Think of the digestive enzymes in your gut that break down massive complex food molecules into tiny water-soluble products so your body can easily absorb them. So we know what they do in theory, but to truly understand how they do it, we're moving to Section 2, the active site's high specificity. Let's actually step right onto the factory floor and watch an enzyme in action. To really set the scene here, you have to imagine a completely chaotic fluid environment. According to kinetic theory, any fluid at a physiological temperature is just full of energy. The heat creates this intense molecular vibration. Water molecules are violently and entirely randomly colliding into everything around them. This causes what we call Brownian motion, which basically means larger molecules, like our enzymes and the target molecules they want to work on, are constantly being pushed and shoved around in this crazy, chaotic, vibrating dance. Now, right within all this chaos, the enzyme has a highly specialized docking station. We call it the active site. And this active site is incredibly fussy. It has a highly specific physical shape and very exact electrochemical properties. Because of all that chaotic molecular vibration we just talked about, random molecules are constantly crashing into this active site. But here's the kicker. If a molecule hits the enzyme and it doesn't have the exact reciprocal physical and chemical shape, like if it is not the perfect lock and key fit, it literally just bounces right off and wanders back out into space. No reaction happens, nothing. But then, amidst all those random collisions, the perfect match comes along. This brings us to Section 3, the enzyme-substrate hug. So when that perfect target molecule, which we call the substrate, finally hits the active site, it doesn't just sit there like a rigid key and a lock. It actually triggers something really cool called an induced fit. The active site of the enzyme beautifully changes its physical shape just a tiny bit to wrap around and bind that substrate perfectly. It's kind of like the video game character Pac-Man closing his mouth right around a pebble, creating this perfectly tailored, snug enzyme-substrate hug. Let's move to and see how this builds into the core mechanical sequence. First up, our substrate randomly collides and binds to the active site. Second, we get that induced fit where the enzyme hugs the substrate nice and tight. Third, the enzyme goes to work actually stressing the chemical bonds of the substrate. And finally, the newly transformed product is released. Let's zoom in even further on step 3 of that process for Section 4, stressing the chemical bonds. I mean, the hug is great, but how does this actually force a chemical reaction to happen so ridiculously fast? Let's use a specific catabolic digestive enzyme as an example here, sucrase. Sucrase's entire job is to break down sucrose, which is just your standard table sugar. Now, sucrose is a disaccharide, meaning it's basically a glucose molecule and a fructose molecule bound together. When the sucrose substrate fits into the sucrase enzyme's active site, the enzyme physically twists and pulls it. It puts massive, literally immense physical stress specifically on the glucosidic bond that's holding those two sugars together. And because that bond is now under immense strain, it becomes highly vulnerable. This sets the stage for a process called hydrolysis. Hydro means water, and lysis means to break apart. So that stressed chemical bond easily facilitates the addition of a water molecule. This water smoothly snaps the bond right in two. And just like that, the complex sucrose has been instantly broken down into two new, highly soluble products, a separate molecule of glucose and a separate molecule of fructose. Both are now totally ready to be absorbed right into your bloodstream. Boom, the reaction is complete. Let's zoom back out slightly for section 5, the infinite loop, and see the immediate aftermath of this beautiful microscopic event. So here is an absolutely crucial rule of biological catalysts. The enzyme itself is never consumed. It's not used up, and it is absolutely not permanently altered by the reaction. The sucrase enzyme just drops the glucose and fructose products, and it is instantly free, perfectly shaped, and ready to bite down on another sucrose substrate. It's an infinitely reusable machine. Now, what's really interesting here is the sheer scale of what this reuse actually means. A single enzyme doesn't just do this once or twice. Because it is completely unchanged by the reaction, a single enzyme processes thousands of substrate molecules per minute. Thousands! That is exactly how the presence of even a tiny amount of an enzyme accelerates chemical breakdown by millions of times, faster than what would happen naturally if you just left that jar of sugar water sitting on a table. But, as incredible as this machinery is, it does have a flaw. That brings us to section 6, when the system breaks. These perfectly shaped, complex 3D globular proteins, they're incredibly fragile. Remember that chaotic molecular vibration we talked about earlier? It is entirely dependent on temperature. At low temperatures, the fluid is sluggish. The substrate rarely hits the enzyme, so the reaction is just really slow. But as your body warms up to its optimum physiological temperature, right around 38 degrees Celsius, the vibration peaks. You get maximum kinetic hits and perfectly efficient reactions. But if the temperature goes too high, like if you experience extreme heat, the chaotic vibration becomes so incredibly violent that the whole system completely breaks down. We call this breaking process denaturation. Extreme heat, or honestly, even a severe shift in your body's pH, like becoming way too acidic or too alkaline, violently distorts the complex folding of the enzyme. The physical shape and the chemical properties of that active site are just permanently ruined. And this is an irreversible process. Think of it like cooking an egg. Once that clear egg white turns white and solid in the pan, it never goes back. The substrate can no longer fit, and that microscopic machine is permanently dead.
Interestingly enough, in clinical medicine, doctors actually use this biological fragility to their advantage. Deep inside your organs, your cells are just packed with specific intracellular enzymes doing their daily jobs. When tissues are severely injured or die, say during something like a heart attack or liver failure, the dead cells actually break open. All those highly specific, microscopic enzymes spill out into the extracellular fluid and wash right into the bloodstream. By running a simple blood test, doctors can look for the presence of these specific spilled enzymes to pinpoint exactly which hidden tissues or organs in your body are damaged.
It really is amazing to just stop and consider that every single process in your body is dictated by these tiny, specific shapes colliding randomly in the dark. Our ability to digest food, build muscle, and simply stay alive relies entirely on these fragile microscopic hugs happening millions of times a second. Which leaves us with a pretty profound thought to ponder. Given how perfectly balanced this whole system must be, what actually happens to the entire biological factory when just one specific type of enzyme stops working entirely? It's a question that drives a ton of modern medical research, and it really highlights just how delicate the chemistry of life truly is.